Welcome to our short documentation showing the complex yet fascinating topic of cell division. Today, we will follow Group 2 as they create solid wire models of mitosis and meiosis to help us visualize these biological processes. The group has divided tasks into equal members with the mitosis group consisting of Cabiles, Escander, and Mexico, while the meiosis group comprises Cabusora, Halig, and Lasagnas. Let's get started with mitosis. The model was created using 6 cutouts of white pad paper to represent the cell. In interphase, prophase, and telophase, the cutouts were circular, while metaphase and anaphase required these papers to be in different shapes to represent the state of the cell. In metaphase, the cell is elongated as the chromosomes are located in the metaphase plate. At the same time, the anaphase shows the formation of the cleavage furrow in the middle of the cell. Additionally, the red and green wires represent different sets of chromosomes. Before we dive into mitosis, the group has made a model of the cell before the S phase. In this stage, there is only one sister chromatid per chromosome, represented by a single green and red wire. This model shows us what chromosomes look like before DNA replication occurs. In prophase, after DNA replication, the chromatin condenses into visible chromosomes. Here, our team is using green and red wires to show sister chromatids. Next is metaphase, where chromosomes align neatly at the cell's equator. In anaphase, sister chromatids are pulled apart toward opposite poles of the cell, separating the green and red wires. Finally, in telophase, 2 new nuclei form, and cytokinesis splits the cell into 2 identical daughter cells, both with 1 green and red wire. This is similar to the interphase model.
Similarly, creating the visual for meiosis started with cutting pad papers into circles, intending to mimic the cell. Like in the visual representation of mitosis, the chromosomes are represented by green and red wires. The visual depicted 2 divisions of meiosis continuously, with their respective stages labeled accordingly. Meiosis 1 begins with a diploid cell, wherein during prophase, the DNA replicates into sister chromatids. The visual depicts the substage leptotene as chromosomes start to condense to its familiar X shape. The chromosome binds to a homologous chromosome, forming tetrads or a group of 4 sister chromatids. Although not explicitly shown, the substage pachytene, also known as crossing over, happened during prophase 1, symbolized by the change in color of 1 P arm per sister chromatid. During metaphase 1, the homologous chromosomes line up at the equator, attaching to spindle fibers. The chromosomes in the visual are then pulled to opposite poles as they reach anaphase 1. Meiosis 1 ends with 2 genetically different haploid daughter cells, each with 1 chromosome and paired sister chromatids. Meiosis 2 begins with similar stages, eventually ending with the sister chromatids of each haploid daughter cell separate to opposite poles. After cytokinesis, meiosis 2 ends with 4 genetically different haploid cells, each with a set of chromosomes. The group wrap up the visual presentation by securing the wired models onto an illustration board. Arrows and labels further highlighted each division and stage. The meiosis and mitosis models were visually separated on the illustration board with a green wire and submitted with the group members' names on the other side.